Well, good afternoon, everyone. And I get to say good afternoon as I'm in Nairobi, Kenya right now. My name is Mitchell Warren. I'm the executive director of AVAC, and more importantly, for today, the outgoing president of Global TB Stakeholders, a critical part of the TB Alliance. Thank you so much for joining us, whether you are in your early mornings or afternoons or evenings. And I can see from the list of participants that we have people dialing in from all over. And it really speaks to, I think, the collective passion and interest in accelerating the gains, as you see on the slide here, around ending TB everywhere for everyone. I have the great pleasure of having been the president of Global TB Stakeholders for the past 6 years. And I'm delighted that in this meeting I get to turn the reins over to a great friend and colleague. Before I do that, I wanted to reflect when I joined as the president 6 years ago. It was at a very different time, to say the very least. I joined, and the stakeholders met actually on the margins of the union meeting that took place in India in late 2019. And we all know what has happened in the last 6+ years. Certainly, a new pandemic spreading and forcing many of us to work virtually and change the way we worked, how we worked, changed what we worked on. Uh, it also then, in the last 6 years, saw amazing scientific advances in TB and in other global health areas, incredible innovations that still struggled to meet the needs of people everywhere, which is why the global stakeholders are such an important group, as you can see displayed here, reflecting civil society that I'm proud to be part of, reflecting donors, implementing partners, normative bodies, groups who all come together to bring an end to TB as part of a larger global health agenda. Of course, these 6 years have also had as I was going to say as a backdrop, but actually as a forefront, the enormous disruptions of the last year in all of our worlds, in the world of global health. The world of TB has not gone untouched and unscathed by massive changes in the ecosystem. And I'm not going to go into great detail of those now. I think everybody on the line has lived these in varying degrees, and we will continue to live them in the months and years to come. But our goal here today is to really be both um, partially backwards looking to what has been accomplished, but more importantly, forward looking. And you'll hear about that in just a minute. In terms of my own personal journey, for me, as someone who works primarily in HIV, R&D, and delivery, it has been a terrific experience for me to be able to work with the TB Alliance and with global TB stakeholders. In this work and provide a bit of context for me to get me out of my HIV bubble. And while I'm passing over the torch to the stakeholders, I'm delighted that I will continue to be a member of the TB Alliance board because I have learned so much, and I hope that connecting the TB and HIV worlds through my work is a value to the TB Alliance and to the larger TB community. It's certainly been great value to me, and it is with that I hope that great value is now transitioned to a great friend and colleague in Doris Macharia. Um, Doris took over the Elizabeth Glaser Pediatric AIDS Foundation, or fondly known as EGPAF, just last year. So her timing is nothing short of extraordinary in taking over a leading organization working at the intersection of HIV and broader primary care in EGPAF, but also leading EGPAF's involvement in global TB stakeholders. And I'm so thrilled that she was elected to be the new president of TB stakeholders and to join the TB Alliance board in that role. And I'm makes me actually very excited to be able to stay on the TB Alliance board so that Doris and I get to work together, both as EGPAF and AVAC, but also as fellow board members in the TB Alliance. We're going to co-moderate today, but I'm going to turn not only the stakeholders over to Doris in her new role as president, but turn over this webinar for her to introduce, and we'll both be moderating some of the conversations here. But Doris, over to you.
Thank you so much, Mitchell, and thanks for the warm welcome. And good morning and good afternoon, everyone. And it's indeed a privilege for me to join you as the new president of the Global TB Stakeholders. I'm really honored to be taking on this responsibility, and I'm sure lots to learn from Mitchell and many of you. Especially at this moment where the fight against TB demands renewed focus, stronger partnerships, and unwavering commitment. And especially for me across Africa, where the burden remains greatest and opportunities for progress are really most significant. But a little bit about me, and Warren and Mitchell have sort of talked a little bit about myself. I joined the Elizabeth Glaser Pediatric AIDS Foundation as the president last year. Really pivotal time, so, going through the ups and downs of the challenges in global health as we've all been experiencing. But more to that, I'm Kenyan, been, based in South Africa, which is my second home, and now the US is my third home, so to speak, and been working in the area of global health for more than 25 years, and really have a passion for mothers, for children, for families, and especially because, as we think about ending pediatric HIV, ending TB everywhere for everyone is extremely important. What really led me to join, the TB Alliance and really stand up and say, I really would like to be part of the global TB stakeholders, is really at the heart of the mission, the commitment towards the AAA, the mandate of new products that will be adopted, available, and affordable for everyone who needs them. This, for me, I think closely aligns with my own passion and decades of work in supporting healthier mothers and children and families. As I was reflecting in beginning this new role, priorities are quite clear, and I look forward to working with all of you to deepen our collaboration and our global partnerships that really drive progress, but also to increase coordination that accelerates access of these wonderful tools, wonderful innovations and treatments and technologies that can really change the trajectory of TB globally. So thank you very much, all of you, each one of you, for your confidence, for your collaboration. And I do look forward to advancing this work together and strengthening leadership that shapes how we see a future free of TB. So as we think about the next 2 hours, how do we want to spend our time and the interactions that we'll have? This meeting primarily focuses on TB Alliance ongoing pursuit of its one-by-one mission, and Mel will walk us through that as well as other colleagues. That is 1 month of active TB and also 1 month of 1 day treatment for latent TB. Those leading the work at TB Alliance will provide key updates, both in science and its strategy, and show how this work of discovery, development, and driving uptake of Pretomanid and as well as BPAL are really informing that path to one by one, and how this foundation of previous successes can really help to inform and set up for even more future successes. Please rest assured we'll have 2 great opportunities for Q&A. Mitchell and I will moderate those. One is in the middle, just after the first session, where we are going to talk about realizing new revolutions in TB treatment, and one at the end. We really encourage you all to submit your questions throughout this webinar using the Q&A function on the Zoom. And with that, and without further ado, I want to welcome and introduce someone who needs no introduction, Mel Spiegelman, who is the President and CEO of TB Alliance. Welcome, Mel. Thank you.
Thank you, Doris. And well, let me just start by expressing, such profound gratitude to Mitchell for the years of unbelievable service, not only as president of this body, but as he'll continue to be as a member of our board of directors. Our board really steers the organization and plays such a crucial role also in its success. And I really look forward to continued interaction and tremendous contributions from Mitchell moving forward from here, in addition to the thanks for the prior service. And Doris, we've only had an opportunity to meet a couple of times, but I have no doubt you will be fantastic in this leadership role of this association. So welcome, and really look forward to working with you too. I think both Mitchell and Doris have mentioned it. These are truly unbelievable times, and I would say not legitimately unprecedented in global health. But we're all aware of the challenges, but I think we also are in a position to really make unbelievable progress. And it's that prior or the accomplishments to date and the future of what we're looking forward to that we really want to dwell on today. Next slide is a pictorial of the TB Alliance pipeline. And I think it goes without saying right now, this is far and away the largest single pipeline in TB drug development and access that exists in the world today. But what I would like to do is really highlight 3 points of the pipeline. The first is what I just mentioned, that this is far and away the largest pipeline. But I think what's even more important than the size is the fact that now we all together play such a large role along the whole value chain of activities, from early discovery work all the way through market access and really getting any drugs any treatments that we're involved in, at least into the hands of as many patients as possible. And so I think, that ability to feed back from all stages of development and access onto earlier and later stages is truly invaluable. And I think you'll see that hopefully in the presentations to follow. I think summary based, and obviously we don't have time to go through the whole pipeline, but I think the summary for me is, in so many ways, the future has never looked brighter in terms of what we can look forward to improvements in TB therapy. And I hope that you'll see that in all the discussions today. If I could have the next slide. And what you'll see in part today is how we've laid the groundwork for the next wave of really quantum leaps in improving TB therapy and how we're going to attack this. Here, what we just wanted to highlight is for active TB, and specifically in drug-resistant TB, the changes that have already occurred that lay the framework for what we're looking forward to achieving in the relatively near future. We've gone historically from 18 in what was up to 24 months at least of treatment with very toxic therapies for drug-resistant TB, down to 6 months with very tolerable treatments, with cure rates that have ranged anywhere from the 30% to 50% to cure rates now that are in the 90% range. And what you're going to hear today is how the next immediate step will be taking this down to 4 months with the substitution of a novel second generation diarylquinoline for bedaquiline. And then what's really the goal for what we're trying to achieve in the next real wave of improvements is coming down to 1 month of treatment, and 1 month of treatment, not just for drug-resistant TB, but for all forms of active TB, whether they're now considered drug-sensitive or drug-resistant TB, and Eugene will go through that in detail. And in addition, on the next slide, we will also be, for the first time really in the TB Alliance history, tackling latent infection. We really have never felt that we had the proper tools to really attack latent TB with the sort of progress that needs to be made in order to make it realistic to reach the 1.5 billion people around the world estimated to have latent infections. Now we feel we do finally have those tools, and so we're going to move on it. And I think that our goal for latent infection, which you'll hear about, and how we plan to achieve that, is for 1 day, as Doris alluded to, which obviously would really potentially change the entire landscape of what could be done for this huge reservoir of TB infection in man. And finally, I think what's most, next slide, what's most important, however, what we also want to present today is even if we have these therapies, they're approved, etc. The next step, which, we could say is the most important step, getting all of this into the hands of the patients and the people who need it. And to make this vision a reality, is really, I think, in many ways, even harder than doing the research and development. But all of it, every single step in this process is critical, because without achieving all the steps in the process, the work, frankly, is for naught. The drugs need to get, the treatments need to get into the hands of all patients. And that's where especially I want to thank all of you on this call, but also all of our amazing partnerships, because this takes not just a village, but a huge city to really achieve these sort of accomplishments. And for that, we clearly are totally in debt to the myriad of partnerships that we have around the world. And with that, let me now turn it over to Eugene, our head of research and development, to actually go through some of the details of what we've done and how we expect to move forward on the next steps. Thank you so much.
Good morning, good afternoon, good evening, as the case may be. And thank you for joining our call, stakeholders. So as Mel said, I'm going to describe say a couple of highlights in our pipeline. Our pipeline is fairly broad and deep, but let me focus on 2 journeys in particular. And drug development is a journey. And we have traversed this journey with Pratominid, as I'm sure you all know, from its very earliest stages, preclinically, through clinical development, through the regulatory and guideline phase, and on to the access phase that Mel just alluded to. And the journey of Pratominid is not just the journey of a single drug, it's the journey of a compound and the important concept of regimen development. And the partners here were obviously bedaquiline and linezolid, bedaquiline being a novel chemical and new class introduced in 2012 or thereabouts, and linezolid being an older drug but repurposed for tuberculosis and an important partner compound in the development of this regimen we now know as VPAL, representing 3 novel drugs with very little or no pre-existing resistance, sort of by definition, as they are novel classes, the diroquinolines and the nitroimidazoles. And importantly, the concept of showing that these components, the 3 in the VPAL regimen, but in any regimen development, the requirement and importance of demonstrating that they're all needed and traversing that challenge with the collaboration and agreement of regulators, who ultimately are the gateway to the availability of these drugs in late stage development. If you can click to the next slide. So skipping a lot of steps, but highlighting that the development of BPAL was driven by three key studies. The NIX-TB trial that was so transformative in demonstrating that this three-drug regimen transformed the treatment of XDR-TB, which at the time had an exceedingly high mortality and did lead to the approval initially of bertomimir in the BPAL regimen, followed by the XENIX trial, which optimized the dosage or certainly improved the tolerability of linezolid, which in the NIX study was used at the high dose of 1,200 milligrams, given the severity of the conditions of those patients. And then the TB Practical trial, a very important study that was a randomized trial with a standard of care, a control arm, and demonstrating the superiority of a BPAL-based, three BPAL-based regimens, actually, but ultimately the BPAL plus Moxi arm was the one that the WHO recommended for standard of care in essentially all drug-resistant TB, from rifampin-resistant to MDR to MDR with fluoroquinolone resistance, and only XDR-TB, newly defined as having resistance to the components of BPAL, were not included. And so this was the journey of bertomimir, and I just wanted to run through that very quickly to illustrate these are long journeys, and the journey's not over. You'll hear about the access part of that journey from Sandeep and Maria later. But we have traversed all these phases of the journey, and we are now embarked on a second journey with sorfequilin. And that very much builds upon the journey of bertomimir. And so let me describe where we are with sorfequilin, previously known as TBAJ876, but now has been given the USAN name of sorfequilin. And so the story of sorfequilin, that journey starts with bedaquiline. And you can see the similarity of the two molecules here on the left. In the middle panel, sorfequilin basically having the design goal of those attributes shown on the right, which was to achieve greater potency and to try to eliminate the QT prolongation liability of bedaquiline. Bedaquiline, obviously, a critically important advance in TB, shown in the lower left-hand graphic, which is the impact on the disease in a retrospective cohort analyzed by patients without bedaquiline in the regimen and with bedaquiline in the regimen, showing the dramatic improvement in their status and outcomes. But then the graphic in the middle panel and at the bottom there, showing the rise of bedaquiline resistance, which is the inevitable fate of all drugs against all infectious diseases, and therefore the need for constant innovation and development of new and better compounds. And with sorfequilin in particular, those design goals of increased potency and improved safety lead to what we hope will be the outcome, the clinical outcome, which are also listed there, which is importantly to take advantage of that increased potency to further shorten treatment duration, to represent a higher barrier to the development of resistance, and to have the opportunity to actually address bedaquiline-resistant TB strains. And I'll try to touch on all of those and how we hope to demonstrate that in this particular journey. Next, please. So skipping a lot of preclinical work that demonstrated to us that sorfequilin did in fact have these attributes and did in fact meet the design goals. Let me show you the clinical data that has confirmed for us that the opportunity for sorfequilin achieving those targets has a high likelihood. So this is the phase 2 sorfequilin dose ranging study that is also known as study NC009 that you may have seen presented at the Union meeting in Copenhagen in November. And let me just walk you through the key features of this study. So 309 patients with drug-sensitive TB were randomized to these 5 arms. The top 3 arms are the dose-ranging portion of the study with 3 doses of sorfequilin in combination with bertomimir and linezolid, given for 8 weeks, those dark purple bars, and then followed by 7 weeks of HR, isoniazid, and rifampin. And the reason for that was when we started this trial, with all new drugs, before you can advance into the clinic, you have to do a certain body of preclinical work focused on establishing safety margins, and safety concerns that one can monitor in the clinical phase. And at the time, we were limited to 8 weeks of clinical dosing. And so there you see the 8 weeks, but we didn't want to just stop. We wanted to continue and see if, in fact, the treatment shortening potential of sorfequilin could be demonstrated. And so this is how we did it under those circumstances with this hybrid regimen, where patients that had a negative sputum at 8 weeks, which would be known by week 15 in the study, could opt to stop therapy and show you what happened there. Then the other 2 arms here are control arms. The BPAL arm is essentially controlled for the sorfequilin molecule, so a direct comparison of sorfequilin at 3 doses with bedaquiline on the same backbone of bertomimir and linezolid, and then the standard of care for drug-sensitive TB, HRZE. So that's the study design. Next slide, please. So at 8 weeks, which was the primary endpoint, the modified intent to treat population demonstrated the following sputum conversion data, showing clearly a dose response from low to medium to high dose sorfequilin, and the high dose sorfequilin arm outperforming both BPAL and HRZE. And interestingly, that BPAL performed at around the same efficacy as HRZE, not having been studied in that population previously. As you saw on the first slide, BPAL studies have pretty much exclusively been in drug-resistant population. Next slide, please. So that was the efficacy. And on the safety side, we're very pleased to see that sorfequilin did not really have much of a safety signal, in that one did not see dose response in any of these safety categories. And it's not easy to see, but on the left graph, the larger bars are the percentage of any adverse event, and to the right of them, the lower bars are the percentage of events that were considered related to the diroquinolin, either the sorfequilin or the bedaquiline. I didn't mention that the first 8 weeks were in fact blinded with respect to the diroquinolin assignment. So one could be on sorfequilin at any of the doses or bedaquiline, but that portion was blinded, specifically for this safety observation to compare events associated with that class. And again, we don't see much dose response across the sorfequilin arms. And then on the right are the more serious adverse events, either the grade 3 events on the left portion of those bars, and then adverse events. I see. Then there's serious adverse events would be the next bar over, and then the third bar would be adverse events leading to early discontinuation, which happened pretty uncommonly in all the arms. So on the safety side, a very encouraging outcome for sorfequilin. We also did not see QT prolongation in that could be attributed to sorfequilin as compared to bedaquiline. So that design goal of the molecule seems also to have been met in this phase 2 study. Okay, next slide, please. So we've also followed patients now for more than 26 weeks, but here's the complete follow-up in all patients for 26 weeks after the end of therapy, which is obviously the important outcome clinically, is the ability to not only convert sputum, but to durably cure patients without relapse. And so very interestingly, the cohort that's shown on top there represents those patients that were able to stop treatment at week 15 based on their 8-week sputum conversion. So that's a 15-week regimen of sorfequilin for half of it and isoniazid rifampin for the second half. The second half being considerably less potent than a full 4-month regimen of SPAL would be, which is what we intend to test in phase 3. And yet in that, let's call it somewhat weakened regimen, we still only see 1 out of 36 relapses, which is about the same rate as the control arms that represent 6-month treatment regimens. So that signals to us that SPAL at a 100 milligram dose is very likely to be a successful 4-month regimen on efficacy. Okay, next slide, please. So just to recap, NC009, a very critical proof of concept study demonstrating that the 100 milligram SPAL regimen has greater bactericidal activity in that 8-week analysis, and subsequently also in the follow-up, that the body of data that we have gathered from this trial is supportive of SPAL as a 4-month regimen. Safety profile and QT profile very favorable and encouraging. So onward to phase 3 on the next slide. And this is the basic study design. We expect to do a study of approximately 500 patients with a 2-to-1 randomization to the SPAL-M regimen in drug-resistant TB compared with BPAL-M in its standard 6-month duration, with a primary endpoint at 1 year from study initiation with follow-up at 6 and 12 months post end of therapy. So we've chosen to study the drug-resistant population for a couple of reasons. One is this is the higher unmet need population by definition. We think that 6-month regimens are better than 18 to 24, but still a pretty long journey for patients, and believe that the potency of sorfequilin can be leveraged to further shorten therapy. And so along the spectrum of drug resistance, which is shown on the bottom there, we intend to include pretty much the entire spectrum from RIF resistance to pre-XDR, i.e. including fluoroquinolone resistance, which will be a stratification factor for the study. And then on the right, you see we expect to encounter bedaquiline resistance, and that there will be an opportunity to explore the activity of sorfequilin against patients that have bedaquiline resistance that are detected during the course of the study, either from drug susceptibility testing from their baseline sample collections or in the course of therapy in patients randomized to the bedaquiline arm and who have treatment emergent bedaquiline resistance. So there'll be an opportunity there to test whether that increased potency of sorfequilin does in fact translate into activity against bedaquiline-resistant mutants. We know that from the preclinical work that sorfequilin's activity against the typical RB678 mutants is equivalent to the activity of bedaquiline against wild-type TB, and so we have every expectation that the drug will be active. And so there'll be an opportunity to look at that in this study. Okay, next slide, please. So this journey for sorfequilin is just the first portion of the journey. We plan to conduct the phase 3 study starting next year and ultimately register, obtain regulatory approvals, and build the case for WHO guideline incorporation. There's another journey going on that you're going to hear about next, which is the exciting formulation technology that has been so transformative for HIV that fortuitously a number of TB drugs have physical chemical characteristics that lend themselves to formulation technology that results in long, sustained, and durable therapeutic levels. And so that journey you'll hear about from Raj. But if you click, we tend to converge these two journeys into what Mel described as a real breakthrough, which would be a 1-month regimen taking advantage of the potency of sorfequilin in the SPAL regimen and the ability for a long-acting injectable to have sustained therapeutic levels for at least 3 months. So in a construct here where patients are started on the familiar 3 or 4 drug regimen orally to determine that they tolerate it and no adverse events would interfere with a continuation phase representing a single injection of 2 or more drugs. So this is the part of the journey that we're still going to be exploring, is how much do you need in the continuation phase, and ultimately we think based on the data on the clinical side for sorfequilin as well as the early formulation data that these are very readily achievable paradigms that we'll be able to see realized in the next few years. And so we're very, very optimistic that we can get there. But I'm going to stop here, I think. Jess, is there another slide? No. So I'm going to turn it over to Raj. But I would like to thank, obviously, the many participants in our trials and the investigators in our trials, our many collaborators and partners, both individual and institutional, and of course our generous donors who have stood with us for all these years. Thank you. Turn it over to Raj.
Thanks, Eugene. Greetings, everyone. So I'd like to briefly describe the program, the long-acting injection program at the TB Alliance. So the long-acting injections offers an opportunity to drastically shorten the treatment for TB. So at the TB Alliance, we're developing long-acting injections for both latent TB as well as active TB infections. For active TB, the treatment could be initiated with the oral lead-in therapy, just like Eugene mentioned, and this will be followed by the long-acting injections. For the latent TB, that people could be diagnosed and treated on the same day with a single injection of a long-acting injection. Long-acting injections are also for selected drugs, are offering an opportunity for application in other disease areas. Next slide, please. So there are some important considerations for development of LAIs, especially considering that low and middle income countries are involved in the TB inspections. So first of all, we have decided to use established technologies that have been used in approved commercial products. So that way there is no surprises later down the road that the technology is very exotic and it will take a lot of efforts to develop it. We are striving to use simple equipment and an uncomplicated manufacturing process, which is, I'll mention again in the last bullet point here. Third is to have established regulatory pathways. So the agencies should not find this very surprising, and they should be able to guide us through this development of these long-acting injection technologies suitable for the TB patient population. It's very important that the technology should be easy to scale up and transfer to the global commercial partners. And for us to keep the cost of goods down, it is important that the generic pharmaceutical industry adopts it, and that can reduce the cost of goods. And most importantly, the AAA mandate from TB Alliance, that means available, affordable, and adoptable. We are trying to adhere to this principle as closely as possible. Next slide, please. So again, reiterating what was just initially mentioned by Mel and then Eugene. So we are striving for a universal treatment, and this is an opportunity to speed global uptake of next generation treatments. So here we're looking for a 1-month therapy, and here the initiation of the therapy would be an oral regimen with 3 or 4 drugs. They'll be administered for 30 days. No pre-existing resistance in these cases. This will be followed by a single injections of 2 or 3 drugs that I mentioned that are going to be used for the oral therapy. And we hope that they will provide in the humans at least 2 to 3 months of plasma profiles or the pharmacokinetic profiles for continuation of the treatment. And that way, in essence, the treatment period is, the therapeutic period is reduced to like 1 month, right after the oral therapy is done. Next slide, please. So here is a target profile for long-acting injections in the regimen for treating pulmonary TB. So indication is going to be first-line treatment of pulmonary TB with oral injections with 4 drugs, followed by long-acting injections of 2 to 4 drugs. The product, we are striving to develop multiple long-acting injections for different agents, 3 or 4 of them. And the volume of these injections, we would like to keep it to be 3 milliliters or less. This is for the convenience of the patients. The duration of the therapy, like I mentioned, it's going to be 1 month with the oral followed by the LAIs. The route of administration is going to be either intramuscular or subcutaneous, depending upon the technology that we select. And then the formulation and dosage administration, we are being very careful that the technologies are simple enough that it can be administered by a local healthcare worker, and no specialized training is required. And any and every component of this long-acting injections will be co-packaged for the convenience of the healthcare provider. Next slide, please. So here is the status update of the LAI development at TB Alliance. The first drug is Sorfecolin. A single administration of a long-acting formulation has given a demonstrated systemic levels that are desirable for a period of 4 months or longer. For Pretomanid, a high concentration solution formulation demonstrates a promising in vivo release profile, supporting the further development of this injectable formulations. And TBAJ587, which is the same class diarylquinidine like Sorfecolin, this is also demonstrated a desired PK profile in the animal models. Next slide, please. So in a summary, a new frontier in TB therapy is within reach. The feasibility has been confirmed. Long-acting injection formulations are technically feasible for a number of drugs that includes bedaquiline, Sorfecolin, 587, Pretomanid, Delasibag, and Rifabutin. It is challenging, but we have accepted the challenge. While significant hurdles in formulation, stability, and manufacturing still remain, these are surmountable with focused efforts. And the promise outweighs the hurdles. The transformative potential of LAIs to significantly impact and improve treatment adherence, simplify care, and accelerate TB elimination makes this a critical and achievable goal. Now I would like to hand it over to Nader to take a look at our TB Alliance portfolio of drugs.
Good morning, good afternoon, good evening, and thanks everyone to join this call. I'll try to take you through just some highlights about the discovery portfolio and how it will impact the development of very short treatment ultimately for TB. I think you've seen this slide before. Mel opened the call with this portfolio. And just to reiterate what Mel was saying, this is the largest portfolio of TB activities, if you will, in TB. What I want to focus on here, your attention, is the discovery portfolio, the three columns on the left. And this is really critical for us to have a robust portfolio of preclinical programs that could then develop into compounds and products that could then transition into the development pipeline, as you can see on the right-hand side. And that's what we have been able to achieve over the last few years. And as you can see, our development pipeline is quite robust. And we've demonstrated that capability by first introducing Pratominid, and hopefully next, as Eugene described, Sofeconlin, which hopefully will be able to replace bedaquiline for a much better regimen for a shorter treatment. So beyond Sofeconlin, what we're really hoping to do is the next phase would be to, we've replaced bedaquiline already, but next we wanted to replace linezolid in that regimen. So next slide, please. And to do that, we've really started to look at all the various pathways that are really key to the survival of Mycobacterium tuberculosis in the infection, and some of them are listed. And as you can see, or as I will point out, the first one is the number one is the electron transport chain, which is really what bedaquiline and Sofeconlin target. The cell wall disruption is very commonly known. The protein synthesis pathway is represented by rifampicin, for example, and linezolid as well. But what we were really interested in is what are the next pathways that have not been explored at all in the past and could potentially replace linezolid. One of them is the central carbon metabolism, that's number 3. Which is really critical for the bacterium. Once you deplete the energy that's produced in the bacterium by the electron transport chain, number 1, the bacterium really shifts into that pathway to generate energy. That's one of the early projects that we have ongoing to be able to discover a new compound in that pathway. But just as important, more importantly for the near future, is the number 4, the protein degradation pathway that we have targeted. And that has 2 components to that. It is really, the pathway is really designed to get rid of, if you will, unwanted proteins or damaged proteins. And it's also very critical for the survival of the bacteria. So we've really targeted that pathway. And if you go to the next slide, we've now identified a new compound that could potentially replace linezolid in the regimen, and that's CLIP-2157. That compound targets one of the components of the proteolytic machinery, and that's the proteolytic component that actually digests the proteins. This compound in the backbone of Sofeconlin and Pratominid significantly increases the sterilization activity. These are all in the preclinical models that we have. And it is as sterilizing as Sofeconlin, Pratominid, and linezolid, as well as Sofeconlin, Pratominid, and moxifloxacin. The addition of this compound also significantly increases the activity, the sterilizing activity of Sofeconlin, Pratominid, and moxifloxacin. We in our preclinical model we have absolutely no relapse after even 8 weeks of treatment in the model. This compound is currently being evaluated in non-GLP toxicology studies, and following that, we will initiate IND-enabling studies to then hopefully go file an IND to then go into clinical development. Next slide, please. So what's our ultimate goal here? Our ultimate goal, as I think it's been said a few times by Mel and Eugene and Raj, is really to develop a universal regimen with novel approaches that really can get the timeline or time frame of treatment to 1 month or less. And if you go to the next slide, what we believe as one key approach beyond developing novel antibiotics that we will continue to do given our portfolio is to target the host and the immune system. And if you look at the left side of this slide, we are now looking into targeting a number of pathways in the immune system, such as targeting the inflammatory component. And those are, for example, the neutrophil infiltration into the lungs, also enhancing the immune response, strengthening the immune response, or improving the memory responses from the immune system. And also ultimately, and preferably, as I will describe in a little bit, is to disrupt the granuloma on the bottom left, if you will, where the Mycobacterium tuberculosis resides, to disrupt that structure and also allow other antibiotics to penetrate more readily into the granuloma to have a bigger and better effect and therefore shorten treatment. So our near-term goal is to really identify 1 to 2 novel therapies, adjunctive therapies, that could then take us into a 1 to 2 months therapy. And in the long term is really to achieve a shorter than 1 month therapy for TB. If you go to the next slide, it's just a quick overview of this slide and the next slide, where a quick overview of how we're approaching this. The first is a partnership that we've developed with the EU consortium called the ITEMIC, and this is to develop adjunctive TB immunotherapies, and we're targeting a number of pathways and targets in the immune system to be evaluated in preclinical studies. We also, at the same time, formed a scientific advisory board back in 2023 to really seek input on the whole strategy, and that really led us to develop a multi-pronged strategy to target the granuloma, as I mentioned. And as a result of that, we formed a collaborative partnership to advance several hypotheses that were agreed with the scientific advisory board as well. If you go to the next slide, and this consortium that we formed, we've put a proposal together, and we were awarded the Department of Defense grant back in July of 2023 to really develop this approach and the strategy. And this has 4 components. First, evaluating a number of different mechanisms of actions in our mouse model of TB. And I will mention that a little bit briefly in the next slide as well, but this really started with applying artificial intelligence through collaboration with a company called Invenio AI to select the targets and pathways that could be important in really harnessing the immune system to get rid of or to accelerate the killing of the Mycobacterium tuberculosis. We then select the most promising the therapeutic modality, and ultimately we would like to then test this in non-human primates, because that's the model, that's the best model that would be that is available actually to be able to understand the effect of an immunotherapy in preclinical models. Following that, we will then gather all of our data, integrate all the data through some modeling, and then which would hopefully help us develop tools to predict the clinical efficacy of the adjunctive therapy. And then following that, we will develop a strategy to rank and prioritize compounds for the best potential clinical application, and then establish a critical path then to feed that back into discovering newer targets and newer pathways and newer therapeutic modalities that we will then test in preclinical models in vitro and in vivo. You go to the next slide, please. And I mentioned that, I mean, we also are trying to adopt as many new technologies as possible. And one of the key technologies that we are looking into and we are applying is the artificial intelligence. I already sort of touched upon the Invenu AI collaboration that we've had. This has been very critical for all of our approaches in immune therapy in selecting the targets, to then to find therapeutic modalities for. And the interesting component of that effort was that all of the pathways and the targets that we've identified have actually already identified molecules that are either being evaluated preclinically or clinically, so that it was very easy for us to then select those compounds, make those compounds, and then start evaluating them in the various models that we have through the two collaborations that I already described, the ITEMIC and the Department of Defense grant that we have with the consortium that we've formed. And then on the right-hand side, we continue to apply artificial intelligence into other components of how we do preclinical discovery. One is to really start looking at novel starting points or novel starting molecules to then optimize in discovering various new antibiotics in the pathways that I pointed out that are really critical for us to pursue. And this has been done with a number of collaborations such as Relay Therapeutics or ChemRx. And then finally, we continue to look at artificial intelligence to not only use that in the optimization of the molecules that we are actually working on, but also in finally the reduction of cost of goods. And this is a collaboration that we've had through the Gates Foundation with ChemAI in specifically right now in looking at alternative ways of synthesizing sulfeconone to see if we can reduce the cost of goods and have a better approach to generating large quantities of material for commercial access. I think that's my last slide, and thank you very much for your attention, and be happy to join the Q&A.
Thank you so much, Nader, and Raj, Eugene, and Mel. Thank you so much for your really great presentations and really great detail in terms of where we currently are with a number of these very homegrown compounds. So thank you all so much. Now, colleagues, you've all had a lot on the scientific side of how TB Alliance is approaching and making real tangible steps towards the realization of the 1x1 vision that Mel introduced us to. You'll soon be hearing more about the delivery side of how the vision is being realized. But I think we want to take a small pause right now and give you all a chance to participate in a discussion by asking questions. So really want to encourage you all to just keep the questions coming. We'll do our best to address as much as we can. And those that we're not able to get to during this initial first round of question and answer, we'll do our best to address those in the second part of our question and answer session. So just to kick us off, this question is directed to Mel. What are the plans to make sulfequilin available to other research consortia and groups that might be interested in evaluating it in combinations other than SPAL? Over to you, Mel.
That's a great question. And obviously, the more work that we can get done with Sofecolin and through, and show its utility, the better. So yes, without going through the details of all the groups that we're in discussion with, we're in discussion with many groups. All of this needs to be done under very rigorous circumstances in terms of making sure that we want to develop regimens. That was clear with BPAL, and it's clear with Sofecolin. What we don't want are drugs simply to be used as add-ons to failing regimens. That's how we got into the resistance problem. But the answer to the question is, by all means, yes. We are fully engaged with multiple potential partners on other regimens.
Thank you. Thank you so much, Mel. And maybe just turning to Raj, you did speak a lot about the long-acting injectables. What are the plans to study acceptability, tolerability? We know little of how patients will accept a new injection, and we may need tons of qualitative data about how it to support and how it can shape, at the end of the day, access. Raj?
Yeah, Doris, that's an excellent question. So from a preclinical perspective, we are running animal studies to see how the tolerability of these injectables is going to be on a very large scale. In addition to that, Sandeep may mention this to you, but as well as a number of organizations in a consortium are doing acceptability studies with the different the groups. And they're trying to go to the patient populations and the advocacy groups, and large number of surveys are being conducted. And this is absolutely right. If this is not acceptable, then why spend so much money? So a lot of effort is being done gathering this information.
Yeah, thanks so much, Raj. And maybe just a quick follow-up on that. The current long-acting formulations is with single drugs. Are there plans to administer 3 or 4 different injections, maybe simultaneously, or just one injection? And also, maybe you can also address how this may pose a risk of maybe resistance. Over to you, Raj.
Currently, I wish I had the technology to combine all the injections into one, unfortunately. But I'm optimistic at some point of a time we should be able to do it. No, currently, the plan is to inject them one injection at a time. So hopefully when the patient is done, goes to the clinic after 1 month of oral treatment, at that point of a time, the administrations of injections can be done.
If I may, just to add to that, I think there might be a little confusion on terminology here. When we talk about single injections, we're talking about a single physical injection, but we always will be giving multiple drugs, either orally or injection. There's no monotherapy in TB, as we all know. And so by single injection, we mean multiple drugs injected at a single session, and rather than a single drug only.
Yeah, thanks, Raj, and thanks, Eugene, for that clarification. And maybe Eugene, can you talk a bit about safety and tolerance of Sofecolin? What long-term sequelae data do we have? Obviously, are there less than what is in standard care? Maybe you can talk to that. Thank you.
Yes. So first of all, as I alluded to, preclinical safety always goes ahead of clinical safety. So with preclinical safety in rodents and dogs, we have studies that are 6 months in duration. So we're well ahead of the clinical dosing duration. We always follow patients for long-term safety and tolerability. As I mentioned, typically for a year after the end of therapy. These are drugs with long half-lives. So I didn't mention that about Sofecolin, but like bedaquiline, these are drugs with long half-lives, not quite as long as bedaquiline, but still long enough that follow-up for months is appropriate. So far, we have only the data from NC009, and in the follow-up, in the treatment duration and in follow-up, we have not detected a safety signal that's unique to Sofecolin, nor a trend in terms of increasing doses, generating more of a particular kind of adverse event. But we will continue to monitor, and then for phase 3, we'll have many more patients than the about 180 that we have studied in NC009. So the safety database will at least double. Yeah.
Thanks, Eugene. And perhaps talk too a little bit about the development plans for sutezolid. Are they still in phase 2 trials? Is that correct? And what are sort of the next steps there? Thanks.
Yeah, thank you for the question, Vim. Yes, so the oxazolidinone story is not over. I think that sutezolid is very much in contention as a potentially better oxazolidinone. We all know of the pluses and minuses, the pros and cons of linezolid, and there are multiple efforts to look at an improved oxazolidinone, as well as Nader mentioned, to take an entirely different approach to protein synthesis. But for now, we don't feel that the data for sutezolid compels us to substitute sutezolid for linezolid, but we're very, very actively looking into that. That is one of the promises of sutezolid. There are emerging studies, I would say, that are going to be informative. And then there are other oxazolidinones that are in the pipeline as well, including a couple of our own. And we're actively always surveying the landscape for new compounds in the pipeline. So that's the oxazolidinone story from my perspective.
Thanks for that.
Actually,
yes, yeah, go ahead.
Doris, I wonder if I could just jump in. Maybe
Yeah.
I know we're going to move on to access in a minute, but I wonder if any of the four of you want to comment. There was actually an article in Devex today about a comment from Jeff Graham of the State Department talking about the next big wave of innovation, and talked about TB specifically at a congressional hearing yesterday. And as both Doris and I come from the HIV world, we spend a lot of time talking about lenacapavir, the injectable, and Jeff Graham said something to the effect that they're looking for the lenacapavir for TB. I don't know that he meant specifically an injection, but I wonder if any of you have thoughts as guidance to State Department and to the field. Where is that next big innovation bet? Not necessarily with the TB Alliance, but in the field, if you were responding to a request for an idea.
So maybe I can take that, because just not coincidentally, I'm in Washington, D.C. as I speak today.
Hence the tie. Washington's a tie city.
Yeah. For a TB Hill Day that occurred yesterday, where Jeff was one of the speakers at the main panel with John Green and Darren Burton from CDC. And after that, I had the opportunity to actually meet with Jeff for an hour in his office. So, kind of hot off the press, actually talking to Jeff Graham about these potential activities that might ensue out of the U.S. Yes, Jeff is very interested in learning of new innovative approaches. We certainly spent a fair amount of time talking about the 1x1 initiative, the use of LAIs. He's very familiar with the HIV story with lenacapavir. This is an evolving situation, and I think, as I'll wrap up later, I think it's highly, really positive. Jeff is very interested in innovation. The U.S. government is really, I think, interested in innovation. Obviously, we all would wish there were more resources being dedicated. That, is just the reality. But I do think within State Department, within CDC and other elements of the U.S. government, we're beginning on a new footing for, how to approach it. And I think TB will be much better situated than it has been in the past.
Thanks so much, Mel, and also for being generous to share in terms of just your discussions that are underway. And obviously, we are all waiting for that magic bullet, but we all know the reality of this work that we've been doing over the many years. So thanks so much, Mel, for just sort of wrapping up the this Q&A session. There's still a number of questions for our panelists, but we need to move to the next session. And I want to hand it over to Sandeep Juneja, who is the Senior Vice President of Market Access at TB Alliance. And we need to pivot to the market access elements and see, how we are translating scientific progress to impact and how all this is working out. And we will get back to the questions that you've all posed. Thank you so much. And over to you, Sandeep. Thank you.
Thank you. Thank you, Doris. And great to be speaking again with the members of the Stakeholder Association of TB Alliance. And thanks for joining us. And I know I see some of the names. It looks like it's pretty late where some of you are. So really appreciate your taking time for this meeting. As Mel said, it's really the work that we do. It's cutting edge. It's really path-breaking, but its utility depends on how soon the medicines that we develop get into the hands of people who need them. So that's what I'm going to speak about, touching very briefly on the next slide about where we are with Predominate. As of end of last year, 2025, the number of prescriptions worth of Predominate that was shipped globally is about 80% of the demand. Now, actual usage on the ground may be lesser. We don't have, as of now, visibility of that. But what we do know is the shipments from the manufacturers we have licensed, and that totals up to roughly 80% of the global demand for MDR-TB treatments. And cumulatively, roughly about 250,000 courses of Predominate has been shipped since the beginning in more than 110 countries. Now, many of you would know WHO guidelines recommended programmatic use of BPAL-M in December 2022. So this has been largely achieved in the last 3 years. So which is really one of the fastest uptake of a TB medicine in recent times. And what we are aiming for, and the question we are grappling with, next slide, is can we do that next time in a much lesser time? Like maybe we should be aiming for 1 year. There's no science to this, but we think making people wait too long is not really ideal given the life-saving and life-altering nature of these medicines. So 1 year is the target. But what will keep us motivated to keep working towards is reducing that time even further. Like, can we do that in 1 month for even further innovations that are in the pipeline? Or why not just 1 day? So the day it's approved, elsewhere, it's approved everywhere in the world, at least in large parts of the world. So that's what is keeping us motivated. That's what we are working towards. And we have, next slide. And we are seriously working on this because we have a very good signal from countries, from potential users about this approach of 1 by 1, especially pan-TB LAIs. Right? We did market studies early on to make sure the investments and research that are happening are justified. And what we do know is a 4-month SPAL regimen for DER-TB would be acceptable to the market, and countries would be willing to adopt that. So the 2-month reduction in duration is seen as a very positive move in DER-TB treatment. For DS-TB treatment, it'll take, what we've understood from market studies is it'll take more than a 2-month reduction to move the needle. you need to get to 3 months or below, and perhaps that is one of the reasons why we don't see uptake of a recent DS-TB regimen that was developed. And we also have initial indications that a 1-month long-acting injectable-based regimen that Eugene and Raj spoke about would be highly acceptable, and there's a high willingness to adopt and pay for that regimen and to switch to that regimen when it's available. Next slide. And we will be refining and improving on the access approach that we have followed for B-PAL as we prepare for S-PAL and the long-acting injectable-based regimens. And I wouldn't go through this slide in detail, but at a high level, while in B-PAL we had a great regimen, we knew it would make a big difference, but we engage with countries and users early on to understand through very systematic and scientific market research to understand which attributes of the regimen will be most relevant for them. And more importantly, what do countries need to change to successfully implement the regimen? That's important because then you can start working with the countries to lay the groundwork for the introduction of the regimen. And this needs to start well in advance. This cannot happen once the regimen is ready or approved. And as my colleagues have explained, TB Alliance's AAA mandate is very clear. The regimens that we develop need to be affordable. So we ensured a very low starting price for Pretominid, which was 60 to 80% below the access price of the other new drugs that have been approved recently. But we also demonstrated that even at that low price, there was a value proposition and cost effectiveness of the regimen in countries, which is critical for them to take decisions to switch to a new regimen. But we didn't stop at that. We also proactively did market shaping for the regimen by arranging a volume guarantee to make sure the price dropped at the point of scale up, not waiting for scale up, but doing it proactively in anticipation of scale up. So, and the third thing is working very, very closely with countries to generate whatever evidence was required at country level. Now, we do know the Nix-TB, Z-Nix, and TB Practical regimens were somewhat limited in geographic scope where they happened. And a lot of large countries do need local clinical evidence. So we had to very quickly get operations research or pilots done in those countries or carry out health economic studies. And that early engagement is one of the reasons why you see that swift uptake of B-PAL. But that was done through a very large number of local partnerships and a few global partnerships. And finally, funding is critical to make this happen. And again, really, we're very grateful to donors who stepped up to see that gap and fund us to make sure that these projects happened at country level. Specifically, the Korean Development Agency, KOICA, and followed by the Australian agency, DFAT, who were, apart from all other donors who support our work, but they critically supported some of this in-country work. Next slide. So the result of that is that we've got access projects in a large number of countries that you see represented on this map, accounting for nearly 70% of the global TB caseload. And the countries where we have worked either have coverage of greater than 80% or have imminent plans to exceed that 80% coverage of B-PAL, B-PAL-M in their respective countries. among new patients who are put on treatment. So yes, the investment in access, enabling access at country level, really works, and that's the proof that we are generating. Next slide. And these are the lessons that we will be applying from BPAL, BPAL-M into SPAL and LAIs. Is field evidence can help, and these are some of the key lessons, of course. I mean, there is a lot that we have learned from this launch, but field evidence can help reduce lag between approval and national policy. So knowing that rather than waiting for approvals and then generating operations research data or data from pilots, we are encouraging high burden countries to participate in our phase 3 program for sulfecolin. So those conversations are going on with a large number of countries. And if it succeeds, it'll be very unlike what has happened in the past for a new drug for TB. We are assessing in parallel implementation feasibility of these new regimens. And we are doing that. We are going to be doing that in the phase 3 trials that are planned through acceptability studies. Cost effectiveness is critical, no matter proving that cost effectiveness is critical. And rather than, again, in the past that we had done it sequentially after the trial, we are planning for those studies during phase 3 to understand really what are the costs that people who undergo treatment bear. And so we can understand really whether the regimens will be cost effective or not. And if they are not, we can take corrective action rather than trying to figure it out after approval. And finally, perhaps most importantly, it's a joined up approach that works. You've got to work with communities. You've got to work on approaches that are led by countries, and we cannot simply parachute in and do that. So we have a large number of initiatives in which we engage with countries. We work through countries and communities, and thankfully we have that infrastructure in place, and we'll continue to expand that and strengthen that. And over the next coming few years, as SPAL and the LAI regimens come to market, those approaches will come handy. I'll give you an example of 3 approaches which are based on communities and countries that we are following. For the first one, next slide, which is a community engagement that we have for access. It's an initiative called Fast Track the Cure. It's a community-based advocacy, demand creation and monitoring initiative. We have worked in 9 countries. I think 5 countries are continuing. Currently, we have projects ongoing. We worked with partners and utilized social media tools for awareness building, demand creation, and monitoring. These 9 country partners have additionally, through their networks, created reach in another 30 countries and engaged over 70,000 community members. That makes this approach very fast and scalable. And needless to say, highly cost efficient. So currently, this approach covers countries, high burden countries, which account for about 50% of global MDR TB burden, and there are plans to grow it even further. And for the next couple of examples, I'll loop in my colleague, Maria Dyachenko, who leads those initiatives. Maria, over to you.
Thank you, Sandeep. Let me briefly highlight the next slides some projects TB Alliance is supporting to ensure rapid and broad implementation of BPAL and future treatment regimes, one of which is PeerLink, which is a knowledge hub established by TB Alliance back in 2024. It is hosted by Tropical Disease Foundation in Manila in the Philippines and supported by the Disease Prevention and Control Bureau of DOH of the Philippines. So PeerLink is a peer-led initiative designed to transform traditional, high-cost and time-intensive technical assistance models. And the aim of PeerLink is to provide more efficient, accessible, and sustainable support, equally available to both high-burden and low-burden countries around the globe. What PeerLink does is it connects experts which really have the actual practical experience of implementation or using the innovative treatments with countries seeking to adopt new treatments, providing support that is highly customized to country needs, and it's scalable beyond BPAL, and it's now actually includes all the WHO recommended treatment options beyond BPAL. Till the date, PeerLink created a global network of peer countries through engagement or providing assistance to 46 countries globally within almost 2 years of 18 months of the establishment. And some of those countries now actually become trainers to others, cascading the knowledge and experience gained through interactions with the PeerLink Knowledge Hub. And PeerLink is further strengthened by establishing the Global Technical Expert Council, which is the next slide. This is a global network of TB clinicians and experts launched in 2025, last year, to specifically consult on difficult cases for drug-resistant TB care worldwide. GTEC supports countries in adopting and optimizing novel drug-resistant TB regimes, responding to complicated cases, which are not standard cases in the routine practice of clinicians. It manages queries and addresses real-world technical challenges addressed by countries. It is now operates with a kind support and kind contribution of 40 experts from 18 countries globally, and it's growing. So hopefully we'll have the representation of most of the regions and most of the countries which have the high TB burden globally to address the queries in the most effective way. It explicitly functions as a case-based and consultative mechanism, and it enables experts review, peer exchange, and problem solving across different country contexts and geographies. While PeerLink and GTEC provide expert support for complex implementation challenges, the next slide talks about the Upskill TB. The Upskill TB is a digital learning tool. It's a platform which builds the sustainable clinical knowledge and confidence needed for broader health workforce at scale. So it is the platform, the digital learning platform developed by TB Alliance in collaboration with the Respiratory Programmatic Implementation and Research Institute in Indonesia and supported by Indonesia Ministry of Health. And it aimed to strengthen the healthcare worker capacity in managing drug-resistant TB on the ground. This platform was designed to address the persistent training gaps that limit scale-up of new regimes. It practically beyond the major urban centers. So the clinical capacity building is now established in the key treatment centers, but this tool helps us to take the knowledge beyond the urban centers to those areas which really need the knowledge retention and clinical capacity building. And this is done in very cost-efficient, scalable, and repeatable digital way. Upskill delivers expert-led modular courses. It is currently focused on DRTB and BPAL mainly, but it is planned to be expanded to other treatment options and obviously the new treatment regimes developed by TB Alliance in the future. It combines the video lectures, it combines the interactive case studies, quizzes, discussions, forums, and all the other approaches designed to support practical real-world application of knowledge through the wild scale. The platform supports multilingual learning, and it has the flexible access, enabling healthcare workers to learn anywhere, anytime. Even if there is a restricted access to internet, materials can be downloaded and actually learned and started in whatever time is appropriate for the healthcare physician. So with this, I would like you to check more on the Upskill TB at the upskilltb.org. And Sandeep, I will pass it over back to you to speak about the impact of TB Alliance's work.
Thank you, Maria. So as you can see, we are using approaches that are scalable, that are technology-driven, and in these times when resources are scarce, that's the kind of approach that you need. So moving on and speaking about impact very briefly. So we wanted to look at these path-breaking regimens that TB Alliance has developed. How much impact has it created, and how much impact will it create in the future? And we collaborated with JHU to develop a model to understand that. On the next slide, I'll show the result from that model. To date, we estimate about 11,000 lives already being saved by BPAL-M in its early days, with savings of roughly $100 million. Over the course of next 10 years, we project a little under 200,000 lives be saved, with 314,000 incremental treatment successes, and a global saving of over $1 billion, with nearly 5 million DALYs averted. So this, of course, does not factor in SPAL or LAIs, which would change this equation. This just is based on BPAL-M. But directionally, this tells us a lot. It tells us that investment in TB research, investment in new medications doesn't only save lives, but it's also value for money. Next slide. And that, I think, will be my final slide. Not much to speak here, but I wanted to share with you a couple of independent assessments and acknowledgements of TB Alliance's work on impact creation and access. The first is under publication. It's a case study developed by the Geneva Graduate Institute on TB Alliance's work on orchestrating access, working with, as Mel said, a village or a city comprising partners around the world to ensure access to TB Alliance, to BPAL-M. So it should be published in the coming months. We'll share that with you. And the other is, again, a shorter case study included in WHO's Country Preparedness Handbook for Introduction and Appropriate Use of Antibiotics, which talks about how BPAL-M was rolled out in keeping stewardship principles in mind. So with that, I'll hand it back to Mel.
Great. Thank you very much. Yeah, I'm conscious of the time, and I want to make sure we have at least some ample time for further Q&A, and thank you all for the questions. And I just want to make two overriding points. One, just to share with everyone, as I had alluded to earlier in an earlier comment, I'm in Washington today still, because the last couple of days were World Hill TB Day in the United States, in anticipation of World TB Day towards the end of the month, I believe on March 24th or so. And I just want to share with everybody the enthusiasm here amongst TB activists, patients, and others is the highest I've ever seen it, in terms of just the number of people involved and really the lofty goals that people really now share for moving the TB agenda forward. And I think for the first time, really, that I at least have been involved in TB work, I think we can now truly look at and also predicated on the progress from all of our colleagues working in TB vaccines and TB diagnostics. When we look at all of this together, now is the time to truly chart a roadmap for TB elimination. Because I do believe with all the progress that now sits on the horizon and the difficulty, but the true possibility to look at genuine TB eradication, not just as an advocacy tool, frankly, but as a real goal within our lifetimes, certainly, and much shorter than that, to utilize all the progress being made and to actually truly get to a point of eradicating TB. So let me just stop there because I'd urge all of us together now to really look at that as a concrete plan that we can all put together as a TB community and truly chart the course to move in that direction. So with that, I think let me turn it back to Doris, I believe, for hopefully we have another 25 minutes for a Q&A.
Thank you, Mel, and thank you, Maria, and thank you, Sandeep. We have a number of questions. I just want to thank you all for just the continued interest and just the questions that are coming through. Mitchell, Warren, and Mitchell and I will sort of tag team on this one because there are quite a number of them. But I'll just start us off. Maybe for you, Sandeep, some of the access initiatives that we've discussed today could possibly be leveraged for additional TB technologies or even technologies for other indications, right? Is there a vision or a plan for leveraging that capacity outside the TB Alliance technologies? Sandeep?
Absolutely, Doris. And we are we focus on one thing whenever we create a platform is how can we leverage this not only for TB Alliance products, but for other products too? So look at PeerLink that Maria spoke about. It started with BPAL, but we've expanded it to all other MDR TB treatments. So PeerLink is now, and we saw that gap when we were running training programs in countries. They came up and said, great to have a training on BPAL, but what about the niche regimens? Like the BDLLC regimen or NTB regimens, who's going to do that? So we immediately expanded and we started providing those trainings. There's a platform we didn't speak about today, the health economics platform. You know, the state of play is you need a health economic study done today in a country. You engage a health economic specialist who comes in, does the study. Sometimes that assessment could cost several $100,000. So we developed a platform in-house with Swiss Tropical and Public Health Institute, which we now replicate in every country, and it doesn't cost anybody anything. So if there's a country that wants to get health economics on TB treatments done, we can do that for them. But we are discussing with colleagues in other PDPs to expand that now to other indications outside of TB. So absolutely, that's our standard playbook. Whatever we develop should be scalable and useful in other areas too. So thank you for that question.
Yeah, and thank you, Sandeep. And before just teeing it up for Mitchell, question to Nada. I think this is definitely an area of my own interest with respect to AI. Just going back to your discussion and your facilitation of that session, are you considering if and how the use of AI can accelerate the speed of getting pipeline of products to active use? And also, and in general, just in realizing the one-by-one strategy that we've been alluding to this morning. Thank you.
Well, thank you for the question. Yes, in discovery, we're using AI in a variety of modes. Mostly right now, as I mentioned earlier, in discovering or at least identifying critical targets and pathways to target the immune system. But in a day-to-day affair of drug discovery, we are now starting to use AI in helping us design molecules based on properties that really are critical to then progress the compound into non-clinical development and IND filing. So, I mean, these are the sort of safety profile of the compounds and the pharmacokinetic profile of the compound and the efficacy of the compounds. So AI is now being used in a variety of our programs in collaboration with partners, accelerate the discovery of the best compound possible to then file an IND.
Thank you so much. And actually, it was a great question and answer to kind of look at a question that came in about flipping back to access. So to you, Sandeep, there's so much talk, and we ended the last session when I asked Mel about innovation, so much talk about innovation and new ideas. And a question, Sandeep, as you work so closely with so many ministries of health, is there a risk of innovation fatigue that we've just seen the shift to BPAL? When we have these new next generation regimens, and especially now given the fiscal and political challenges, are you getting a sense that people may say, you know what, slow down, I just transitioned, I can't deal with another change? And are we in the R&D field going, we never go too fast. We always think it's too slow. But is there a risk of overwhelming the system? And if so, how is the TB Alliance thinking about bringing people along this innovation journey so that it meets the moment?
Right. No, absolutely. We are seeing that, and BPAL-M seemed to be a no-brainer when it was introduced, but countries had to make a lot of effort to switch. Even if it was simpler, sometimes it's an effort to unlearn something that you're used to because it's complicated, and there are specialized people who know how to deal with it. So that was an effort, and but we were surprised when we got a very positive signal from countries that they were willing to switch to a 4-month SPAL. So we parsed through the data that we were getting to see what matters to them. And the 2-month reduction in duration mattered to them. What also mattered was that SPAL actually is not really changing things a lot. It's a known platform, which is BPAL-M, and you are continuing, you're simply improving the diarylquinoline element of that, and you're taking away some really important pain points over there. Even though you don't see a lot of cardiotoxicity in the field, but you still have to really test the patient every month. You have to bring them back and do that ECG. You will likely take that away. You are most likely able to snip away 2 months of linezolid, which is a problem for many countries, which is where at least we have observed, the countries have observed a lot of peripheral neuropathy happens. So you're snipping that away. So you're solving those problems realistically. But if you come up with a remarkably new approach, okay, a completely new novel class within 2 or 3 years of introducing BPAL, that's where we saw there was going to be resistance. So this is going to be a significant improvement without disturbing the status quo too much and solving some of the key pain points. So that's why. But LAI is so transformational that we heard from countries that kind of a change, absolutely, we are willing to make that kind of accept that kind of a regimen, even if it's a big change. Hope that answers your question, Michelle.
It does, Sandeep, and I think it really does speak to the need to be in constant collaboration with ministries and implementers so that we're not catching people off guard or coming with the next big thing. Maybe I'll ask one more, and then I'll flip it back to you, Doris. There's a great question from a friend and colleague in the chat about any data related to Pritomanid in children, and then more broadly with next generation products, the role of children, adolescents, and pregnant and breastfeeding women in participating in clinical trials to make sure that we are not seeing the historic lags and delays. I'm not sure who wants to take that.
I can take that.
Oh, go ahead, Eugene.
Yeah, sure. So we are trying very hard to get Pritomanid moving along faster on the pediatric front. I would say, frankly, pediatric drug development is more complex because of, obviously additional safety concerns, but also regulatory challenges, that there's not really harmonization across agencies, and that makes it a little bit more arduous. But we are making progress. We have nearly completed the third or fourth of a several-step process for Pritomanid. We have done the preclinical work to pave the way for Sofequin, and we have worked with the EMA pediatric governance process to develop a pediatric development plan. For pregnant women, we have done the necessary preclinical work to establish safety to the extent that one can in preclinical studies, and we'll be taking a more, let's say, liberal approach to pregnancy within our trials. Previously, we would have patients would have to discontinue the trial if they became pregnant. But in our phase 3 study, we're planning to allow patients who become pregnant on study to continue on the study and generate data that way. But we still have a long way to go. We're very aware of the need there. And so we're chipping away at the access issue for those special populations.
Good. Thanks, Eugene. And as Sandeep was sharing earlier about the sort of transition to BPAL, just for you, Eugene, can you talk to what is known about cross-resistance between Sofequin and Bedaquiline? I think that would sort of help to sort of tie up the discussion that was earlier facilitated by Sandeep. Thank you.
Yes, so Sofequin has the same target as Bedaquiline. It's the same class of drug. The mechanism is the same. It's just more. So if you think of resistance as an animal jumping over a fence, Sofequin is a fence that's 10 times higher. If you measure it in the traditional way, which is determining the concentration of drug that you need to kill the bacteria. That's a pretty conservative estimate, because the test tube methods don't necessarily reflect what's going on in people, in infected patients. If we account for other differences between Sofequin and Bedaquiline, we think that fold difference in potency is more in the 20 to 25 fold range. In other words, the fence is maybe 20 to 25 times higher. So that's why we believe that the increased potency will make it harder for resistance to develop. And resistance develops, it's related to tolerability, it's related to the duration of regimens, which really challenge adherence. If you've ever been treated for an infection and been given a 10 to 14 day course of antibiotics, which sounds trivial, most people actually don't take the 10 to 14 days. They forget, they get better, and they say, I think I'm cured. And so that's why with chronic infections that take months to treat, the adherence is the opening that the bacteria have to develop resistance. And the higher the barrier, the more forgiving, you could say, the drug is for a momentary lapse in adherence, which of course all gets solved by long-acting injectables, right? You take the adherence out of, unburdens the patient
from having that responsibility. So the mechanism is the same. It's just a higher barrier. The resistance signatures that we know for bedaquiline will be the same for serfeculin. It's just that they won't matter as much, we think. But all that remains to be proven.
Yeah, but
Yeah.
also I think and Eugene, maybe you'll speak a quick minute, that even existing bedaquiline resistant strains clinically, the possibility that serfeculin would overcome that resistance and something that we are looking into how best to test for that clinically.
Yes, so right. So we've in animals, we've shown, as I said earlier, that serfeculin's activity against bedaquiline-resistant TB is the same as bedaclin's activity against non-mutated TB. And so that's why we believe that in the clinic, as we encounter bedaquiline resistance in our phase 3 trial, we'll be able to overcome that. But again, remains to be proven, but the theory is sound and the preclinical data are fairly substantial.
Great. I'm going to hand it back in just a minute to Doris to close us out. But I thought perhaps one last question in our final couple of minutes before we do that, which is open really to any or all of you. And that is a question about the way forward, and particularly thinking about the engagement of TB survivors, of civil society, of advocates. And we know in many ways, once we get to the delivery part of the equation, access, communities can often get very animated and very involved. We see that in HIV. I think we see it at Sandeep in what you're presenting. It's often harder to engage in further upstream science. And what you've presented collectively over the last hour and a half or so is an incredible, from early discovery through clinical development and through access. And I would just love to hear any of your reflections on what more might we as a collective do, and what might the TB Alliance do to help bring TB survivors, advocates, and civil society in meaningfully engaged ways. That's certainly how we became involved at AVAC in our partnership with the TB Alliance and good participatory practice. But what more might we do, and what do you see the future for that intersection as? And happy to have it come from both R&D colleagues as well as from our access colleagues.
So maybe I'll take the first stab at that very quickly, because it actually came up in a discussion from a survivor, a TB survivor, at the panel yesterday on World TB or Hill TB Day here in DC. And really the point that was made is countries choose policies, their country policies, which then really decide what happens to a great extent with TB patients in each country. Those policies, I believe, can be very much influenced by the activist community, by the patient community, by all of the constituencies that you mentioned, Mitchell. And we have to generate that enthusiasm and really make sure that that capability is there on a country-by-country basis, because frankly, as we've seen through the leadership in certain countries, if we can get policy change and enthusiasm at the highest levels of government, that's when we can see true change in terms of what happens with TB in each country. So that, to me, would be really the plea for all activists and engaged communities, is really constructively to engage with governments and really push for that sort of true policy change.
And maybe, Michelle, I can add on the access side of things. A pivotal development that happened in HIV in the early days was the community, as you'd know, rallied around and worked energetically on blowing the stigma around HIV. And not that it's gone, but it's really very different now. Treatment is much simpler. It's easier in HIV than it used to be some 20, 25 years ago. I believe in TB, we are now catching up with HIV as far as management is concerned. And with the simpler treatments that have been introduced and are being developed, time is now right to start talking about TB differently. That it is an infection which is treatable, and you didn't do anything wrong to get it. So I think that's where communities can really play a big role, and that'll help us solve a big problem we have in TB is the treatment gap. In MDR TB itself, you've got 400 odd thousand people who could be infected, but only about 150,000 are treated. So if we can blow the stigma, we can find the missing people and help them get to treatment. And communities can play a major role. So maybe that's something for the communities to take on, and we are happy to partner with them on that, along with so many other colleagues who work in the space.
Thank you, Sandeep. Where has the time flown? It just sort of went past so fast. Thank you so much to you, Sandeep and Mel, for taking that last question and wrapping us up quite nicely with regards to the questions. And just colleagues, very quickly, you've all had just really exciting work that is currently underway, and the approaches today that we have more opportunities to remain in dialogue with you all throughout the year. And this is going to be ultimately important. We want to, oh my goodness. Hello?
Yes, I can hear you, Doris.
Okay, all right. I think it is, I think I sort of froze. Apologies for that. But just quickly, just to say that we're planning to host a couple of additional web events this year, so that where we can have a really good deep dive on some of the areas that we've talked about today, and also additional topics. Please, can you see this QR code? Please scan it. We want to hear from you. We want to know what topics you want us to cover in future web events, so that we can tailor make this and ensure that we can address your needs, answer questions, and be able to interact with you throughout the year. We'll also send a link via email as well. So please, if in case you're not able to scan this right now, that will be coming shortly via email. We'll also be sending a summary and recording of this meeting in the coming days. And as part of that, we will address the questions that we were not able to answer today, because there were quite a number of questions which we are very excited to take on, but I guess we just ran out of time. So I want to just say thank you all so much. I want to say thank you to Mel and your leadership, colleagues who spent time today to sort of walk us through the innovations and just tell us the exciting news of what is currently available, and also how we can continue to play a role as stakeholders in ensuring that we can enhance access, affordability, and availability of these compounds to ensure that we can eliminate TB. So thank you all so much. And thank you, Mitchell, for just your wonderful friendship, and for just a wonderful transition as well. And look forward to engaging with you all in the coming days, months ahead in this year. Thank you all very, very much, and have a good day.
Cheers, everybody. Thank you.
Bye.